Good afternoon, everybody, and welcome to our meeting, where the aim is to wrap up the pilot research project that were part of the development of Neurocenter Finland. So the funding period is now ending at the end of this year, and we are now currently establishing the permanent center and having negotiations going on. So most likely we will start our permanent operations in the beginning of year 2021. The pilots have been really essential in our preparatory phase because we have gained experience of how to boost a collaboration between different researchers and also between researchers and companies. And today we will hear what the pilots have achieved during this 2-year funding period and how we are then going on with the collaboration. So I will start with a short presentation of Neurocenter Finland, its goals and future prospects, and then we move on to hearing about the 6 pilot projects, speeches from their coordinators. So welcome to this very interesting evening with neuroscience. So here is today's schedule. And as I mentioned, after these welcome words, we will then start with the actual pilot presentations, and we will finish approximately at 4:15. Okay, so do we see the slides? Okay, so maybe you could help with.
Let's do it like this.
So why Neurocenter Finland? We know that today in developed countries, brain diseases are by far the most costly and biggest health burden. It has been estimated that during their lives, every second woman and every third man will face a serious brain disease, meaning that practically everybody knows in their close people somebody with this disease. Brain diseases are also very costly. It has been estimated that the cost of brain diseases, including the memory disorders in elderly people, but also mental health problems in working age people, their costs are at least twice as big as the costs of cancer and cardiovascular diseases together. And due to the population aging, it's most likely that the costs will be even higher in the coming years. Neurocenter Finland is now being established to cope this challenge, and our main aim is to boost collaboration. So we think that collaboration is the key for success in fighting the brain diseases. Our mission is to achieve better brain health and also new care for brain diseases by 2 ways. First, and most of all, by strengthening collaboration between researchers and companies, and also by promoting innovations in this field. We aim to be an internationally recognized player in the coming years, promoting Finland's competitiveness in the fields of neurosciences. We have two main aims. We want to promote cross-boundary partnerships between academic companies and also third sector actors, and by that way promote Finnish neurosciences globally. Secondly, we want to make ideas into innovations. We want to make the mind setting in the field of researchers so that they would be more willing to consider ideas, innovations already in the research phase. And we also want to give essential support in this field. And finally, we want to promote the Finnish research infrastructure, the availability of health data. We want to act as a mediator between the data producers and researchers and companies. And overall, we also want to collaborate with other centers of expertise in personalized medicine, and by that way promote Finnish know-how. So, what we are hearing today is about the pilot research projects. During this establishment phase, we got a task from the Ministry of Health and Social Welfare asking us to launch a call for pilots who would be beneficial in this development phase. We got 54 applications in a quite short notice, and out of these we made 6 different baskets, and out of these baskets we then succeeded in getting 6 very high quality collaborative projects. We hear today about stroke from Nina Forss. We hear about Alzheimer's disease from Mikko Hiltunen, and we also want to boost infrastructure, so we are part of imaging biobank pilot, and Lauri Parkkonen will tell more about this project. Rare diseases are very important, especially in the Finland setting, which has a genetically isolated population, and these rare diseases need very special attention. Brain tumors are also presented today by Joonas Haapasalo, and finally we look at the secrets of multiple sclerosis. What have we achieved during this almost 3 years preparatory phase? We have now 12 organizations who are willing to establish a Neurocenter during the coming years, consisting of 7 universities and 5 university hospitals. In these 6 pilot projects, there are altogether 37 organizations involved and also 8 companies. We provided the seed funding for the projects, and during these 2 years they have been able to collect more funding, almost 6.3 million euros, and out of these 1.5 coming from abroad. So this is one of the main aims of Neurocenter Finland. We want to boost collaboration, and we also want to get more funding from outside Finland to support research and development in neurosciences. So we are really happy with these results. But Neurocenter Finland also provides non-monetary value for science. So from the pilots, we have learned that they have been able to strengthen existing collaborations and create new exciting networks. They have been able to increase multidisciplinary collaboration, taking also other fields like computational neurosciences involved, and they have also been able to improve the visibility of Finnish neuroscience both nationally and internationally. So with the pilot project, we have really been co-creating tomorrow. But the work is just beginning, so we need further collaboration to be even more successful. Today, research in Finland is already very high quality because we have these unique databases. We have also patients who are really willing to participate in research and who are willing to donate their samples for research. But now we need to invest even more so that we could reach a very high, even leading position worldwide. And now we want all of you to participate in this collaboration, to join our network of Neurocenter Finland. Our success critically depends on all of you, researchers and other network. So thank you for participating today. So we will have really exciting speeches coming. Please also notice our web pages and our social media channels. So we want to provide you with all newest up-to-date information in Finnish excellence in neurosciences. Then I would like to introduce our next speaker, Nina Forss from Helsinki University Hospital, will present challenges in diagnosis and prevention of stroke and their pilot projects. So, Nina, please, the floor is yours.
Okay, hello everybody. I am Nina Forss and I'm presenting you the results of Pilot 1 together with Professor Ville Kolehmainen from University of Eastern Finland and Adjunct Professor Jukka Putala from Helsinki University Hospital. I share my presentation and I hope that you see it easily. Just a moment. Here we go. I hope that you can see it. Can you see my presentation?
Yes.
Yes, we can.
That's nice. So our pilot is actually divided into two different projects. The other one is developing novel technology for monitoring of hemorrhagic stroke patients, whereas the other one is focusing at cryptogenic stroke in young adults. And just to remind you, I put this in a presentation mode, it's easier to follow, that there are two types of stroke, and the symptoms are very similar. We have hemorrhagic stroke that occurs as weakened blood vessel rupture, and there's a bleeding into the brain tissue. Whereas ischemic stroke is due to lack of blood flow, as there's a blood clot or embolia that stops the flow of blood into a certain brain area in the brain artery. Our first project is focusing to this hemorrhagic stroke, how to monitor these patients, and our other project is focusing at this ischemic stroke, and Jukka Putala will tell you about it a little bit later. I'm now going to tell you with just a couple of slides, why do we need a novel technology for monitoring of these patients? Here you can see our truly multidisciplinary teams working with this project. these are the PIs of these teams. And I think, correct me, Ville, if I'm wrong, but I think that you all guys knew each other and had collaborated earlier, actually forming a Center of Excellence of Academy of Finland in inverse imaging. But with the funding that we got via Neurocenter Finland, we at the hospital were able to join this team of top experts. And I have to say that this has been very fruitful, exciting, and very fun to work in this project.
Yeah, that's correct.
That's nice. So when we are talking about monitoring of hemorrhagic stroke patients, here you can see a CT scan of a patient who has a very small hematoma in the right side of the brain. Oops, now something happened with my, let me see. Now you see it again, I hope. So that there is a small bleeding in the brain. So if now the circumstances remain stable, the prognosis of patient is quite good. However, it is quite possible that during the couple of hours, the first 24 hours, the bleeding continues or restarts. And then the situation is quite different. The hemorrhage that is shown here as this white area and the edema surrounding it occupy space inside this bony skull, and that leads to increased intracranial pressure. There are compensation mechanisms that can decrease this pressure, but at a certain point they don't work anymore. And then eventually the brain doesn't fit the skull anymore, and it is squeezed out of the head, actually, most typically downwards towards spinal cord, and that causes eventually the patient's death. So the only way we can monitor at present this size or the growth of hematoma is to clinically evaluate the patient. We have to figure out whether the vigilance is the same. That means if the patient is getting drowsier or even unconscious, and if the neurological symptoms are getting worse. It sounds quite simple, but it's not, because these findings are quite subjective and it is prone to errors. Most importantly, they are non-specific. These are very seriously ill patients. They are often intubated, and that means that we have to give sedation to these patients, and we are losing all clinical hints that we have in order to follow this hemorrhage. The only way to do it is to move this critically ill patient to a CT scan and check what is the situation. Well, timing of control imaging is also an issue. If the image is quite okay now, what about after half an hour or 1 hour or even 1 day? How often should we take these images? We take repeatedly CT scans, which is always a risk for critically ill patient, and it also increases the dose of X-rays in these patients. Similarly, when we try to follow up our ischemic stroke patients, we face the same problem. We have very effective treatment nowadays for acute ischemic stroke, like thrombolysis therapy or mechanical thrombectomy. But the intracranial hemorrhage is a feared complication of this recanalization treatment. And the only way to detect hemorrhage in this area of ischemic brain is to follow the symptoms of the patient once again. Here you can see a CT scan of a patient who is developing quite large infarction in the right side of the brain. But if the patient gets worse, we really don't know whether this is due to increasing edema, like in this case, or if there is a hemorrhage in this same area. And our treatment strategy in this situation is quite different, and we should get the understanding what is going on in the head as soon as possible. So what we really need is a bedside online monitoring method that would give us information about the status of the brain. Even in any other patient in intensive care unit would benefit from this kind of technology. For example, major cardiac surgery patients are in risk of having ischemic stroke due to embolia or bleeding because they need to have quite a lot of anticoagulant medicine during the operation. And as you can see from these real life images, there are so many different tubes and systems, monitors surrounding these intensive care unit patients that it's even difficult to find the patient between these tubes and machines. And it is practically impossible to take these patients into CT scan. In the future, I hope, we may be even capable of using this kind of technology in differentiation of ischemic and hemorrhagic stroke from each other. As I said, the symptoms are very similar. So that means that when a patient gets the stroke, nowadays we always have to take the patient to the hospital as quickly as possible to take the CT scan to see whether it's an ischemic stroke or maybe a hemorrhagic stroke. And the treatment is, of course, quite the opposite. We have to start the thrombolysis therapy for the ischemic stroke as quickly as possible to save brain. And if we could separate them already at the field and start this therapy immediately, that would really be a game changer globally in stroke treatment. In this project, the role of HUS Neurocenter is to provide medical expertise wherever it's needed. We produce brain imaging data of healthy subjects and brain hemorrhage patients for the other team members. And when the technology is eventually ready, we start the measurements of healthy subjects and then again after that in stroke patients. So we really need this technology, and Ville, now I'm asking you to tell us whether we are going to get it and where we are going.
Okay, thank you, Nina, very much. Shall you change the slides and I will explain?
Yeah, let's go nice.
Okay, so hello everyone, I'm Ville Kolehmainen. I'm a professor of computational physics at the Department of Applied Physics at the University of Eastern Finland, Kuopio campus. And I will now explain kind of this, let's say, more technical part of the new sensing technology or monitoring technology we have been developing for. And our idea basically is to use, instead of using X-rays, we aim to use electrical currents for probing the brain. And here on the left is sort of a picture of our colleague Samuli Siltanen. It's a 6-year-old story in Helsingin Sanomat. So we have been working on this idea, kind of the basic mathematical theory for quite a while. So this idea was discussed in this Helsingin Sanomat article. So the idea is basically like Samuli is having here, that the patient at the intensive care unit will have an EEG hat, so basically a set of electrodes placed on the scalp. And then instead of connecting the leads to an EEG device, the leads are connected to an electrical impedance tomography device, which injects small alternating currents into the tissues using the electrodes. And here remark that the frequency of the currents has to be more than 1 kilohertz, typically say 10 to 20 kilohertz. So frequencies on these currents on these frequencies are completely harmless to the patient. No ionization, radiation, no harm at all. And then while the system feeds the currents, it simultaneously measures the induced voltages on the same electrodes. And then this voltage and current data is used to reconstruct a 3-dimensional image of the electrical conductivity of the tissues. Similarly, as all the tissues have different contrasts in different MRI imaging techniques in CT imaging, they also have different electrical properties. And here, especially, for example, if the patient has an hemorrhagic stroke, it means that there is an amount of highly conducting blood in the tissues, and this will be seen as a local high intensity area in an EIT image. So the idea is basically very simple, of using measurements of electrical currents and voltages to retrieve 3-dimensional information about the electrical properties of the tissues and use this information to assess the status of the stroke. And as mentioned here, we have been working on this mathematical groundwork for this problem quite a while in the Finnish Center of Excellence of Inverse Problems. Can I have the next slide, please? So during this Neurocenter project, we have been we have designed and built an electrical impedance tomography system for stroke imaging, and the system is shown on the image on the left. And here, this system is connected to a three-dimensional printed head model in laboratory. The reason is that whenever developing a new imaging system, it has to be first tested in vitro with different phantom specimens, etc., before moving on to imaging actual human specimens or even animal specimens. So we are currently in a stage with this device that we can do feasibility studies using kind of in vitro subjects, and we have used, for example, this kind of three-dimensional head model which has electrically realistic model of skull. And then this device itself, as you can see, it's quite modular and small in size, so that can be very easily fitted on the intensive care unit. I mean, these units that are on both sides of the head model are basically kind of the circuits for current injection, voltage measurements, and those can be placed on both sides of the patient's head on some shelf or support device. And then there is this one kind of a larger unit below the laptop that contains the kind of voltage sources, etc., and then the laptop computer for sort of controlling the measurements. At the moment, this device, well, basically, it also can be connected to a standard EEG hat, so the kind of the sensor end that is attached to the patient can be the standard EEG electrodes that are used for EEG measurements in hospitals. And the status of the device itself is in a way that we are in process of calibration and final testing of this device, and basically are soon in a position to apply for an permission to carry out human studies with this device. And then the image on the bottom shows this three-dimensional head model. The reason I have put this image here is because I will show some preliminary feasibility results using this kind of simple 3D printed head model. Then can I have the next slide? And then this is now an in silico feasibility test of the new approach. And in silico means that it's fully computer simulated. So we have constructed a realistic electrical model of human head based on a CT image, and then used mathematical programs to simulate noisy EIT measurements, and then use the image formation algorithms to reconstruct the images. So this illustrates the idea. So on the top, you can see the CT images. So basically on the left, 2 hours after the onset with slightly smaller hemorrhage, and then on the right with 6.5 hours after the onset with larger stroke. And as Nina explained, currently the only way to monitor this is basically monitoring the vigilance of the patient, and then whenever needed, take him or her to radiology department for CT imaging. So with this EIT-based system, we can basically do the monitoring on the intensive care unit. So then the middle row shows sort of simulated brain conductivity roughly corresponding to these CT images on the top. Remark that they are not from the same head model, but I'm showing the CT images here to illustrate kind of the, in a way, the connection with this current way of monitoring and this new approach. And then on the bottom row are shown the EIT results from this in silico experiment. And then the third image on the bottom row shows the reconstructed chains based on these two, between these two time points. So basically, you can see that the growth of hemorrhage can be seen quite similarly as in CT, but now the physical parameter we are probing is different. In CT, it's the tissue intensity. Here, it's electrical conductivity of the tissues. Can I have the next slide, please? Then here is a laboratory simple kind of a feasibility test. So this is using the 3D printed head model. So basically testing the performance of the device. So again, this would be like a simple laboratory example of a follow-up of ischemic stroke. So on the left, the brain has no hemorrhage, and then after the treatment of ischemic stroke on the right, there would be a kind of secondary stroke in the brain. And then this middle row shows the simple lab model. So kind of here we have just added water in the tank. So basically the conductivity in the initial state is homogeneous inside the skull. And then we have added on the right sort of a small, more conductive inclusion to in a way simulate or mimic the effect the hemorrhage has on the conductivity contrast. And then the bottom row shows the images. So basically the occurrence of the kind of increase in the conductivity intensity caused by the hemorrhage can be detected very clearly on the EIT image. So in summary, it's sort of a just very simple measurement device to probe the kind of brain with electrical currents, and we are in a way hoping that we would get in a way similarly information from the growth of the stroke as we do with CT. The spatial accuracy is not as good as with CT, but I mean it's more important that we would have some kind of a systematic way of detecting the changes in the status of the stroke. Then in future, well, we are aiming for clinical evaluation of this new technology. And basically this Neurocenter funding has been really crucial for enabling our initial collaboration with Helsinki University Hospital team and identifying and clarifying the clinical goals and research problems that need to be solved for making this new technology clinically feasible. And then we did apply altogether funding for this project, and we received 1.3 million from Janne and Aatos Erkko Foundation for this future developments of this technology. And the partners in this project are UEF, Helsinki University Hospital, University of Helsinki, then Aalto University and Tampere University. And the main technical issues in the future development, well, basically EIT imaging is highly sensitive to modeling errors. So basically in the computational model, we would need to know the locations of the electrodes pretty accurately and also the shape of the head. So for this, we can employ different auxiliary measurements to kind of localize the electrodes. And also we are aiming to build a patient-specific monitoring model, and this model can be very conveniently constructed based on the CT data of the patient that is taken always when the stroke patient is coming into hospital. So in a way, that's basically lots of technical work to construct that kind of a personalized imaging model for each, or in a way the pipeline for constructing the personalized imaging model, but once the pipeline is done, it's very easy to employ. And then this system needs to be equipped also with an automated alarm for stroke changes. So kind of instead of looking at the images in the intensive care, this system has to say beep or give some kind of alarm to the doctors and nurses if there are changes in the brain of the stroke patient. And then finally, when we get to the state that we can do experiments at Helsinki University Hospital, it will be very interesting to see what is the sort of, let's say, sensitivity and resolution limits of this technology. These are, we can get some estimates using highly realistic in silico experiments, but kind of the final certainty will be only obtained after the clinical feasibility studies. Thank you.
Thank you, and I'm sorry that we are taking quite a lot of your time, but we still have Jukka Putila to show 4 slides about the project number 2. Jukka, you can present yourself while I'm looking for your presentation here. Do you hear me, Jukka?
Yes, I'm here.
So here we go. Can you see it now?
Good afternoon. Good afternoon. We have slides here. So this part of the pilot project 1 focuses on ischemic stroke in young people, and specifically such patients who have so-called cryptogenic strokes, and that means patients who don't have any elusive cause or etiology for the stroke despite full clinical workup, and the problem is not big. The problem is not small globally, because 1.3 million young patients with ischemic stroke remain without elusive etiology. And when I'm speaking about young people, I mean those who are between 18 to 49 years. So this is a clinical study where we enroll 600 patients from a total of 20 dedicated sites and 600 controls, stroke-free controls for these patients. We utilize standardized diagnostic workup and clinical data collection methods. We collect blood samples for further analysis of genetics, hypercoagulation, and so forth. We have established core labs for reading of key diagnostic studies, including brain MRI and neurovascular imaging, cardiac echocardiography, ECG. We will follow up the patients for up to 10 years. So can I have the next slide? And this project is a multi-center international project, and the base funding comes from the Academy of Finland and 5 university hospital districts in Finland. And Neurocenter Finland has ensured this project's continuum during different periods of funding from other bodies. And we also, with the help of Neurocenter Finland, we have built up a strong network for this and future multi-center projects. And our aim also, besides of doing science here, is to create an internationally attractive research environment within this collaboration. Can I have the next slide? And here I list the funding for the project, which we have obtained so far. I first received a personal grant from Finnish Medical Association, then we received grants from Helsinki Uusimaa Hospital District, and then a consortium grant from Academy of Finland for years 2015 to 19, and again from one funding from Helsinki and Uusimaa Hospital District. And I also received another funding from Academy of Finland to work as a clinical researcher. And then before we received the next consortium grant to continue the enrollment for the study, we applied funding, this pilot funding from Neurocenter Finland, which served as an essential bridge funding between the two consortium grants obtained from the Academy of Finland. So the role of Neurocenter Finland from funding point of view was essential to ensure that we can continue enrollment for the study. So at present, we have, as our target was 600 patients, we have now 530 patients and about 400 controls enrolled. And our target is to complete participant enrollment by the second quarter next year, after we can start fully analyze the full database. Can I have the next slide? So we already have some publications out of the material. We have published 2 papers on study protocols, one for the main study and one for the echocardiography analysis. We have published one paper on left ventricular hypercoagulation imaged in the cardiac MRI. We have one result from advanced echocardiography imaging regarding left atrial function, which was impaired in patients compared to controls. We have another result from advanced echocardiography imaging regarding left ventricle diastolic function, which was also impaired in patients compared to controls. And then lastly, we have a recent result regarding the role of microembedded aura as a risk factor for cryptogenic stroke in young people. And we observed that microembedded aura was a particularly strong risk factor for these kinds of strokes in both men and women, and irrespective of a very common cardiac finding, patient foramen ovale. I think these were our my slides at this point. Thank you very much for your attention.
Okay. So thank you, Nina, Ville, and Jukka for the very interesting presentation and also future prospects. So does anyone from the audience have questions? So if not, I would make actually one question to Nina. So you mentioned that you are, and Ville, you mentioned that you are now planning to move to clinical validation studies in the coming years. So how do you see, what will be the greatest challenges in this, also taking into consideration the new medical device regulations? So are there some very specific issues that you are planning to face, and are there some things that Neurocenter Finland could be assisting them?
Well, if I start, I think that a lot of paperwork has to be done, and I think that we are quite well prepared for it. But any help that can be we welcome any help, because I think that there are a lot of new issues that we haven't been dealing with before, and certainly most of them are dealing with the different kind of contracts and licenses that we are doing. But otherwise, I think that from medical point of view, all the challenges that we are facing are such that I'm quite certain that we are capable of overcoming them. And it's of course very important that we start the measurement with healthy subjects just to test the feasibility in real life. But I don't know if you want to, Ville, comment on the other issues.
Well, I guess this sort of from the technical side, the device design has been done such that all the medical safety issues are taken into account. But I have to admit also that getting into a bit of new waters in this, how this sort of application process from Valvira goes. So I think support or instructions or help in that might come worthy later on.
Okay, so thank you. Then I think we will move to the next speaker. So Mikko Hiltunen, professor from University of Eastern Finland, will present translational research on genetic risk factors of Alzheimer's disease. So Mikko, please, the floor is yours.
Okay, thank you. So hopefully you'll see my presentation soon. It seems that it's still loading that. Okay, can you see my slides now? Can you see my slides and hear? Okay, thank you.
Yes, we can.
Okay, so first of all, I would like to thank the Neurocenter Finland for giving this opportunity to present our project AlzTrans, which stands for Translational Research on Genetic Risk Factors in Alzheimer's Disease. So this is, of course, a multidisciplinary effort which we have now conducted for 3 years together with several partners, and hopefully I'm able to introduce you and present the outcomes which we have received during this project now in this AlzTrans. But before going into the details, I would like to introduce the partners and the collaborators which have been instrumental here. Of course, without the people, this would not have been possible. And the University of Eastern Finland was spearheading this project. But apart from me, there were several people involved. Mari Takalo, Henna Martiskainen, Petra Mäkinen, Anna-Kaisa Haapasalo, and Ville Leinonen, who were all instrumental for executing this project as planned. In the THL, we had several partners there, Mia Kivipelto, Tia Gandu, and Jenny Lehtisalo, who were part of the finger population assessment and the identification of those study subjects from this population. From the University of Helsinki, Professor Jari Koistinaho was playing the major role, but apart from that, Taisia Rolova was participating for this project actively. And finally, from the Turku Pet Center, Professor Juha Rinne and his crew, Ulla Kulmala and Mikko Koivumäki, were basically doing a huge effort in order to be able to arrange the logistics related to this project. And of course, the key collaborators here should be mentioned: Kuopio University Hospital, Auria Biobank, which is the most relevant partner now in our phase once we are entering to the 2021, the FinGene study, the EADB Genetic Consortium, and also the UF's Bioinformatics Center, which all played a major role in these projects. So what was the rationale in AlzTrans? So I will just remind you the idea behind the project. So in the recent genetic and the functional studies in Alzheimer's disease, we have identified several novel risk genes and the variants which are basically involved and play a role in the innate immune system. And this is interesting because obviously it shows that the microglia seems to be a key component in these early phases of Alzheimer's disease pathogenesis. And therefore, given this, we have been kind of thinking whether we should go beyond the p-values there and define these translational approaches and to test these risk gene variants, particularly those rare variants which are having a big risk effect, whether these are usable in different kind of applications which are focusing on personalized early biomarkers and therapeutic targets in the future perspective. And here you see the slide where I have outlined a couple of those major goals and objectives what we had in the AlzTrans in the first place. So indeed, we wanted to see how determine the translational applicability and the feasibility of these novel AD-associated microglia-specific risk gene variants. And here you see in the down right, so you can see those variants and the genes which we identified, which are in the same biological pathway in the microglia. So we have the API3 gene, and the variant there, which is known to increase the risk of having Alzheimer's disease approximately twofold. And then at the other end of this spectrum, we have the protective PLC gamma-2 variant, which is significantly decreasing the risk of having Alzheimer's disease. So, and because they are in the same biological pathway, our assumption was that in focusing on these individuals, asymptomatic individuals having these variants, we could find some insights to the potential mechanisms and targets which can be used later on in personalized manner. Also, we were applying different kind of cell models, disease models, focusing on the microglia. We're using the blood-derived or monocyte-derived microglia-like cells, and then iPS-derived microglia from the same individual. So the idea was to see and evaluate the similarities and dissimilarities between these models, which can be later applied into personalized medicine approaches in different settings. And finally, importantly, because we are dealing with the microglia, we kind of concluded that it would be possible to use a PET imaging focusing on lichens, which are specifically kind of playing a role in the microglia, to see that whether we can detect any kind of major changes in the activity of microglia in the asymptomatic phase. And this was the initial setting, and then putting all things together, we thought and planned so that we will use different kind of neurobioinformatics approaches to get this data together, since we had a lot of global omics-based data which needs to be kind of analyzed in the organized and integrated fashion, and taking into account also the PET imaging results. So this is the practical part. So you can see the outline of the strategy during these years. So we initiated in 2018 in the autumn by focusing on the finger cohort and selecting the individuals based on the GWAS data, what we applied or which we produced from that cohort in the large European genetic consortium. We were able to identify both these API3 variant carriers as well as BLC gamma 2 carriers, and then started to recruit them to this study plan, or this study. This was the hardest part of the game, since we, before that, having these individuals to come into these studies, we needed to have all the ethical approvals and the legislation in order, and that took the most of the time for 2018 to actually get the ethical permissions and the logistics and the recruitment policies in order. But then after this was done together with the THL partners and the University of Eastern Finland, but then we got started and we were able to start to invite these people for these analyses, which I just mentioned, and there you can see what was the idea. So the individuals went to the Turku PET Center, where the sample collection was made for the blood and the skin biopsies. And at the same time, these individuals attended to the PET and MRI imaging, as well as the neuropsychological testing. Subsequently, the samples were sent to the University of Eastern Finland, the blood samples, and then the skin biopsies to the University of Helsinki. So this was a huge logistic effort to execute this as planned. And then after, individual partners were pursuing the analyses as planned. And currently we are in the phase at the end of this year, so that we are able to put together a decent number of individuals which are having these variant form, or then the wild type form to the final analysis, so that we can have the clear outcome measures there. And of course, these were the centers which were participating for this analysis. And here you can see what was the logistics at the Turku PET Center. So it was a 2-day procedure where in the first day, the blood and the skin biopsy samples were taken. Then the first PET imaging part, and subsequently the MRI imaging. And in the second day, neuropsychological tests were made, and the PET imaging for the second ligand was conducted. So unfortunately, the last spring, of course, we had this problem with the COVID-19, and we were not able to assess all the individuals which we wanted to assess, and therefore we focused on this last part for the API3 risk increasing variant carriers, rather than going into the BLC gamma 2 variant carriers. But this is something which we are pursuing now in 2021 by using additional funding obtained from different sources. So what about the science, which is always the most interesting part? So first, of course, we wanted to validate our models. What we are using, of course, Professor Jari Koistinaho has already validated the iPS-derived microglia production, and therefore there were no need for that. But for us at the University of Eastern Finland, the MDMI cells needed to be validated, originating from these individuals having the variant or then the wild type allele. And obviously we were able to, together with the Kuopio University Hospital, we were able to validate these MDMI cells. So we got from the from the with the help of Professor Ville Leinonen, individuals from the KYS to come and get the samples, the blood samples, which were then confirmed by using different kind of methodologies so that they were feasible for assessment in our omics-based and the functional analysis. So definitely these MDMI cells were expressing those microglia-specific markers which we were expecting to see in those cells. So saying that the model for related to the MDMI cells was acceptable and good. So in order to see how these MDMI cells were in relation to these peripheral blood mononuclear cells, so what was the difference, how the differentiation took place. So here you can see a typical volcano plot, what we are using in these bulk RNA-seq analyses. Of course, there's a lot of information, and I'm not going to go through all the details. I just want you to put your emphasis and your focus on these heat plots where you can see in different times, different patches of these MDMIs produced as compared to the PBMC cells. And what you can see here clearly is that we have very consistent expressional changes between these PBMCs and MDMI cells, and that we are getting these expected targets upregulated in the MDMI cells, such as TREM2 and LPL, these typical microglia-specific genes. Also, in the same time, once we knew that the MDMI cell model is operating as it's supposed, so we did the comparison between these variant carriers, API3 variant carriers, and the wild types. And importantly, we were able to identify several upregulated and downregulated target genes, which are playing an important role in different kind of biological pathways in microglia cells based on the previous literature. And this is something which we are currently continuing by having more samples, by having more individuals having this variant or the wild type for the analysis. So this is still ongoing part, but initial results already shows that we have several interesting targets which are already showing the relevance based on the recent literature. We also conducted in these MDMI cells the functional analysis in order to see how well they are operating. And what we are seeing there in these initial results is that these these variant carriers having this 209 variant, so that there are alternations in the inflammation response, as you can see here, after the induction of the stress by using the lipopolysaccharide and an interferon gamma. You can see that those individuals having the risk increasing variant, so they are secreting more IL-6 as compared to the wild type individuals or the individuals without having this variant, as well as the internal control, which is we are using here. And the difference here also is that this individual is much younger as compared to these individuals who are in their 60s and 70s. Also in the survival assay, we are seeing that these variant carrier MDMIs, they are not surviving as well during this 48 hours time span, so suggesting that the survival is affected by this variant. And also in the phagocytosis assays, we are seeing this very clear trend towards increased phagocytosis of different kind of material there in these protective risk increasing variant individuals, both in the vehicle or non-induced situation, as well as the LPS induced situation. And currently we are processing more of these samples in order to get statistically relevant and affordable kind of outcome measures. So what has happened in the University of Helsinki? So Professor Jari Koistinaho is now in the process of producing the microglia cells. So we have these iPS cells established, and now the microglias are coming, and from these API3 individuals as well as the wild type. Also, Jari Koistinaho has already produced the API3 knockout by using the CRISPR-Cas method in order to see how the API3 functions in these functional assays, what we have also used in the MDMI study. And here you can see the differentiation protocol, which they have been established, and also the functional analysis related to the API3 knockout cells versus the wild type, where we are seeing the same kind of outcome measures as what we were seeing in the MDMI, so that there's alternation in the phagocytosis of material there. Also, they are seeing differences in the expression of specific microglia-specific markers there. But this is ongoing, and we are expected to have these results, which then allows us to do the comparison with the MDMIs early in the next year. Finally, this is the situation in the Turku Pet Center, what they have done so far. As you already saw from the protocol, how these samples or these individuals were kind of handled there in the Turku. So they had 2 PET ligands which were applied for them. The other one was the amyloid specific ligand, and the other one was neuroinflammation or glia specific ligand. And interestingly, what we are seeing based already in this initial result is that particularly with this neuroinflammation glia specific ligand, we are seeing a significantly increased binding of this ligand in the lateral temporal cortex of these API3 variant carriers as compared to the H and ApoE matched non-carriers. So this is extremely important information because these individuals are asymptomatic individuals. They are only having this risk variant, and because they've been matched with the ApoE status, it suggests that there's already an activation taking place in the microglia due to this AD-associated risk variant in this early phase of their lives, and gives the idea that the PET imaging would be suitable for measuring already these predictive measurements there in the early phase. Amyloid doesn't show, at least with this number of individuals, yet any changes there. So this is something which we are also pursuing in this project in 2021. Okay, even though it would be really nice to continue with talking with science, so I think that these tangible outcomes and the added value of AlzTrans project is needed to describe. And here are some general impacts, what we have outlined it here, and what we are seeing as a clear added value of having this AlzTrans project. So indeed, we have now established successfully in the multicenter level, this translational medicine platform, which allows us to really put an effort and the emphasis on the personalized medicine purposes. We have tested and validated that this system is working. So we have established the ethical approvals, legal issues, contacts with different biobanks, particularly Auria Biobank in Turku and Helsinki Biobank, recruitment policies for the study subjects, the logistics, all these have been already successfully been applied and to be used in the subsequent studies, which are focusing on similar outcomes. AlzTrans has, at least our point of view, significantly promoted this new national and international collaboration in the academia, but also in the pharma industry, because we have been in touch with the pharma industry, which is interested in pursuing this collaboration with us in other targets. And importantly, we've been now outlining the best practices in the translational medicine, which is combining the basic research clinics and the biobanks. And of course, as being scientists, money is always needed. And we've been really successful in this by having this AlzTrans project as a basis for applying further funding, not with the same idea, but going beyond the existing ideas and the results. And as a good example is the funding from the JPND EU co-funded project in the last year, which provided us 1.5 million euros and combining 4 different centers, including the University of Helsinki, Turku Pet Center, DCTMI from Germany, and Institute of Pasteur Lille in France. So this is a major effort which is now ongoing, and obviously we are able to leverage and further develop all these facets what we have produced and established during the AlzTrans project. Apart from that, our young researchers have been able to get funding from the Academy of Finland for their postdoctoral research funding. So in total, the funding budget so far is more than 2 million euros what we have gained during this AlzTrans period. And importantly, this means that AlzTrans will continue its activities after the end of the official project funding period, which is at the end of this year. So definitely this project will continue, and all the aspects of these different facets will be studied further. And of course, in the scientific impact is important, and as I already mentioned, so that we definitely have identified already potential novel functional mechanisms and targets, and now the time is to really put and test them in larger cohorts, and for that purpose we have now initiating the collaboration with the EADP consortium in Europe, which comprises tens of thousands of AD patients and controls, which allows us to assess these initial results obtained in the AlzTrans project. So from our point of view, the most important thing, if we think about the science, is that we have now established a state-of-the-art research platform, which is unique in the global scale. So at least what we have been looking the literature and knowing the field really well what comes to the Alzheimer's disease, we haven't seen this kind of setting nowhere. So this is a really unique setup what we have, and therefore we are planning really to apply this in the multiple other targets which we have now identified in these global GWAS studies where we have intimately been involved. And obviously this has led to the scientific renewal of these ideas, so that we are moving away from the solely neurocentric view in aiming to understand molecular mechanisms in Alzheimer's disease. This is an important thing. So it's been reshaping our research in all these universities and university hospitals which have been involved there. So I'm really eagerly waiting the outcome measures, which hopefully will be eventually being seen as novel, tangible applications related to the personalized medicine, which can be then integrated to the patient care and the prediction in the clinics and the biobanks. Finally, of course, I would like to thank and outline the support of Neurocenter Finland, because this has been instrumental. All these aspects which and the facets which I went through, these are being originating because of this project. So this is the important thing to remember, that without the support from Neurocenter Finland, this would not have been happened. And this has been very synergistic and active collaboration between the leadership in the Neurocenter Finland and all the players there. And they've been really helping us in the multiple different facets there, which are related to the funding in the national and international level. We have now been able to do that in Finland and in Europe. And then, of course, the important thing, what Neurocenter Finland has been actively been doing, is the dissemination of the AlzTrans concept and the results nationally and internationally. We are really thankful for that, because that has clearly created and promoted the crosstalk between different partners and the funding bodies. And definitely we have now established the links between the biobanks, data lake providers, and importantly also the industry, which is probably in the core of the Neurocenter Finland. And of course, finally, I would like to emphasize this important issue, because it seems that collaboration is self-evident in many cases, but truly in this project, we have now established new multi-center based collaboration in Finland beyond the existing one. And I'm really kind of looking forward these possibilities, what these different kind of projects which we are still pursuing, what kind of outcome measures we are able to succeed in the future perspective. Okay, so there's a lot of, probably I went over my time, but I really would like to thank you all for your attention. And if you do have questions, so I'm ready to answer those. Thank you.
Okay, so thank you, Mikko, for the fascinating presentation and really top quality research that you have been doing with Alzheimer's disease. So we have time for one question. Do we have any from the audience? Okay, then I would ask one question. So, Mikko, you mentioned in the early slides that the possible future prospect could be finding early biomarkers or even therapeutic targets for Alzheimer's disease. So, how do you see the possible collaboration with the future Drug Development Center and Neurocenter Finland in this respect?
Yeah, of course. I mean that now having all these targets, which we are evaluating currently, I mean that it really provides a platform and a setup for doing this kind of collaboration with those kind of institutions which are able to take this to the next level as well. So, of course, we are not clearly on in our expertise in this kind of therapeutic kind of developmental system. So we definitely need some additional help in order to really get these tangible outcome measures, what you mentioned just recently.
Okay, so thank you. Then I think we will move on. So the next speaker will be Lauri Parkkonen from Aalto University, who is the coordinator of National Brain Imaging and Biobank Pilot. So, Lauri, please, the floor is yours.
Okay, thank you, Mikael. I hope you can hear and see me and more importantly, see my screen. Do you see it?
Yes, we can.
Okay, good. Yep, so I will talk about the National Brain Imaging Biobank, which is one of the pilot projects. And in this project, we are trying to address in one way the need for better diagnostics of brain disorders. So as Mikael already pointed out in his introduction, brain disorders constitute a major societal burden. There are direct costs involved and of course a lot of human suffering. But diagnostics is difficult. Every brain is different, so there's a lot of individual variability already among healthy brains, and then it's not always easy to tell a diseased brain apart from healthy brains because of this variability. Also, it's hard to link symptoms and brain states in many cases, and also we have limitations in brain imaging. It is not measuring everything we would like it to measure. It's not available every place, and it's costly quite often. So now what we are trying to address with this brain imaging biobank initiative is that we could aggregate a lot of data and use this, what are known as big data approaches. So we could use them for building a model of individual variability, which we could take that into account then in the derivation of, say, for example, biomarkers for certain brain disorders. And in doing that, we could then apply AI tools or machine learning. So for this project, we built together a consortium, initially of 7 partners. So the coordination is with Aalto University. Then we have University of Oulu and Oulu University Hospital with Vesa Kiviniemi and his team, and also Ville Leinonen, who joined the project after the application phase. So he's also in Oulu. And then together with him, then Ritva Vanninen from KYS. Then Turku University Hospital is also involved. And then HUS. and then the sort of company technology providers, CSC and Combinostics. I will talk about CSC's role more a little later. So the role of Neurocenter was instrumental in this project. Without the support, it would not have happened, mainly because this is more about building a research infrastructure rather than conducting the research itself. And there are actually not too many funding mechanisms in Finland for this kind of activity. At the same time, I have to say that the funding did not nearly cover the costs that we have incurred during this pilot phase. So let's say that we have probably put twice the amount of money into this thing compared to what we got from Neurocenter. But anyway, it was instrumental to get this going. So the aim of this project was to establish a national network for doing functional brain imaging and gathering the data together. And we chose specifically two methods to do that, MEG and then so-called MREG, a fast variant of fMRI. I will come back to that in more detail in a minute. The second aim was to pilot the IT solutions for doing this data bank, so to aggregate data and to analyze it in a cloud-based manner. And the third aim, that was the one directed by Ville Leinonen, so to harmonize structural MR imaging protocols across radiology centers in Finland. So briefly about the brain imaging methods that we are using. So MEG, or magnetoencephalography, that is a measurement of electric brain activity through the magnetic fields it generates. So we have sensitive sensors around the head and can detect the very, very weak magnetic fields due to neural currents. So this is a direct measure of neural activity. It has a fairly good spatial resolution, but most importantly, it has very high temporal resolution, which is about 1 millisecond or so. So it's compatible with the speed of events in the brain. So in many senses, MEG is optimal for studying functional connectivity of brain regions, but that's again a region where we need to have quite a bit of data to establish, say, a normative situations. And this is what the biobank is aiming at. The other method that we chose for this consortium is the MREG, or magnetic resonance encephalography. That is about 10 times faster in terms of sampling rate than the usual fMRI. So with fMRI, you get an entire volume maybe once per second or once per 2 seconds. But in MREG, you get an entire imaging volume every 100 milliseconds. So what that can buy you is that you no longer have to let your data to be compromised by physiological artifacts, such as the pulsation of blood, but instead you can actually study this thing. And this is what this glymphatic imaging is about. So you're looking at the blood circulation and its changes in the brain. And then in the same way as with other fMRI or MEG studies, you can then compare these results between patients and healthy controls. The downside of this approach, or the MREG approach in general, is that the image reconstruction is a pretty heavy computation. It cannot be done directly in the scanner, as is the case with normal MRI or fMRI, but instead one has to use at least a small-scale computer cluster or even supercomputers. And this is where CSC's role is instrumental again. So in this project, the MREG part established an international unique network. There are, of course, fMRI imaging networks in many countries in the world, but to our knowledge, there is no doing this ultra-fast imaging. And the aim of the MREC part of the project is to advance brain diagnostics, resorting to the glymphatic imaging approach. And in Finland, we have a fairly good network. So there are now 10 3 Tesla scanners from the same vendor that have this MREC sequence installed and can run with the compatible protocols. And here are some early results from Vesa Kiviniemi's group in Oulu, showing that by doing this MREC imaging, one can actually see substantial differences between patient populations and controls. So here there are schizophrenia patients, epilepsy patients, and Alzheimer's patients, and they all show quite widespread changes in the glymphatic system. And there are the papers, one of them published, one accepted, and one in submission. And if you want to have more information about this, so then please ask Vesa Kiviniemi, who is probably participating this meeting as well. So then moving on to the actual biobank or data bank. So it has a web portal at the address braindata.fi, so you can just browse there and have a look. You can't access the actual database just like that, because the access, of course, needs to be authenticated and a permission given, even though at this point we mostly have sort of example data in that publicly, or well, not publicly, but in that part of the database. The implementation is already on a CSC system, and so by having it there, so we can rather easily make it available for all universities and university hospitals in Finland. And so here I have a video showing how to use it. So now from that main page, you can browse to the actual database interface. And this is now a video showing that how you first authenticate yourself. So we are using the authentication system by the Elixir project. And for that, you can use your university credentials or hospital credentials. And now if you want to look at the data sets there, so you can set filters, many of them. So you can only get task data sets that have a certain number of data records. They have certain imaging modality, a certain kind of a task, and so forth. So you can apply many filters at once. And then once you have found what you're looking for a study, so then you can go in that and then look at the details, look at the individual recordings. Here you can see that there was MEG and also structural MR imaging of the same persons. And you can see what was the task there. And then eventually then you end up with the information about which datasets could be useful for your study. Now the important thing to remember here is that the actual data is not stored in that database, but it's the metadata. So information about which kind of recordings are there. Now then, of course, the database also allows adding data there. This is similarly a video about how to do that. So you authenticate yourself, of course, first, and then you supply information about the data set. And indicate which kind of a recording, and then you can use your browser to browse the files that are on your computer in the so-called BIDS format. And then the browser application will go and retrieve the meta information from those recordings and send it over to the database. Again, the actual data is not sent. And then it tries to figure out as much information as possible from the file headers and so forth, but then it will ask you to supply the extra information that it cannot determine automatically. right, so then there is one more data set in the database. So what we are still working on is that we are collaborating with THL to define ontologies, and that happens in a context of an EU project about digital biobanks. So now, of course, biobanks have been tissue sample based, so there are physical samples, and then the terminology has not been there for digital samples. And now this needs to be standardized so that one can take maximal advantage of different biobanks. So they need to be interoperable, and for that it's necessary that we are using the same terms for the same things. Another thing that we are looking at with THL is the pseudonymization of the imaging data. The current idea is that THL would maintain the associations between the actual subject identities and then the pseudonyms which we use for encoding the data. And then this biobank system will not have in any place the real identities of any of the dataset owners or providers. So now if one wants to do a register study, so then the first contact point would, once you have found the recordings, would be THL, and they could give you the list of the pseudonyms that you would then look for. But the real identities of the study subjects would never be disclosed to the researcher. Then how to use it, or what to do with it, what to do with such a large body of data. This is just an example. There is no news here because, well, we know that the alpha frequency peak goes down in frequency a little bit when people age. So that has been known for several decades, I assume. But anyway, we can see that in our sample of 650 healthy controls here. What is maybe not so well known is that if we, instead of looking at the parieto-occipital junction, if we look at the areas around the central sulcus, then this slowing is a bit less with age. So if you compare these two, so then the slope of the curve is slightly larger in for the alpha activity from the parieto-occipital junction. Okay, but that was just for healthy people and no big news. The more important use of that kind of data is to derive biomarkers to help in diagnostics. And for that, we took a small set of brain injury patients and made what could be called significance maps of the differences of those patients with respect to the healthy controls in the database. And this is an example just of one of those patients in one location of the cortex, shown by this blue dot here. And this curve here, so although it has frequency axis here, is not a spectrum, but instead it tells you how significantly at each frequency the brain activity in this particular patient differs from the normative sample. So for example, here just below 10 Hertz, where we would have the alpha activity, there is no difference. This is not to say that there wouldn't be alpha activity in the patient, but just that it doesn't differ from the normal population. But instead, then somewhere around 20 Hertz and a little above, so there's highly significant difference in the amount of that activity. And you would have this kind of curves for every location in the cortex. And now what we can do with this is we can train a machine learning model to try to predict or to classify which data sets are actually traumatic brain injury patients versus controls. And we've done that using a support vector machine classifier, using a radial basis function kernel, if there are people into machine learning listening. This is just a ranking of the features. We had altogether 60 features, and then, well, the names are a bit coded here, but anyway, they have some physiological meaning. So you can go back to your data and probably understand why the classifier is able to tell patients apart from controls. And if we now imagine what could be done with such a database, the long-term outcomes could be that we could facilitate this kind of register-based research. We have pooled data, maybe not just from MEG and MREG modalities, but the data could also be connected with other kind of data from the same individuals using this pseudonymization approach with THL, as I explained earlier. Hopefully, this will produce more efficient tools to help in diagnostics, particularly of neurodegenerative diseases. And then, of course, these imaging tools are pretty expensive. There are not that many MEG systems in Finland, 3 at the moment, and also MRI, fMRI, and particularly MREG is not that widely available either. So we of course hope that we could turn some of these findings then into cheap bedside technologies, like having the MEG results inform how to make an effective bedside EEG solution for the same purpose. What these could be, I don't know at this point, but this kind of a framework could provide the basis for building such things. Right, then in the end I'd like to acknowledge, of course, the Neurocenter Finland support for this project and the entire consortium that we had. It has been great to work with such a consortium, but then I would like to specifically thank the team at Aalto University for actually putting the database thing together, doing the implementation, software development, and for this example results and initial results that I showed you. And then also I'd like to thank the group of Nina Forss for providing the TBI patient data. And thank you for the attention.
Okay, so thank you, Lauri, for the excellent presentation. And as we heard, you speak about a new research infrastructure, which is really important. So do we now have any questions from the audience? Okay, we have one here. I will look. Okay, so I think Nina, you have raised your hand, so please, if you have a question.
Yes, I have. First of all, I would like to say to Lauri that this is very important, and I'm fascinated about these primary results. And it should be as easy as possible to add the data to this system. And now, if I think about just practical questions, if I have collected data with the permission from the Department of Neurology, for example, you always have to tell who keeps the registry, which is actually hospital in this case. And even if the data is pseudonymized, not anonymized, we always come to the same question, whether you would need to have permission from each subject in order to take this data to these kind of banks. Do you have any solutions to this? Because I thought that I have, but we still are having problems in other projects.
Yes, yeah, this is not a totally solved thing. So for that reason, this was an excellent question. It seems now that it's possible to have consent forms where this thing is taken into account. How to actually have the wording there, that we are in the process of finding out. And the other important thing is that how much that could apply retrospectively. So to data sets that are already there, so can they be uploaded to this kind of a database? And this is actually quite a bit of work to find out which kind of ethical permits and consent forms would allow the data to be submitted to the database. So that we do not know yet precisely. However, the thing is that because the database is at this point only holding the metadata, so the actual sample values won't go there. So I think the restrictions are a lot milder there because you are not sharing the data. You're only sharing the information about the fact that such data exists. And now then, if someone really then wants to have the data, from you for his or her analysis. So then that person comes to you and asks for the eventual permission. So it's not such that they would just go and download it from some system and then start using it without you knowing. So you still have one point where you can say that actually the ethical permit doesn't allow this to happen at this point, or you would need to go and ask the study subjects again.
Okay, so thank you, Lauri. I think the question that Nina asked is really crucial from the biobanking point of view, and I'm sure that there will be solutions for that in the future. So now we are a bit short of time, and I think we can catch up during the next break. So I suggest we will be back at 20 to 3 according to the schedule. Okay, so now after the short break, welcome back to this seminar. Next, we will hear greetings from the Ministry of Economic Affairs and Employment of Finland, and Development Manager Anni Kaukoranta will give the greetings. So, please, Anni.
Thank you, Mikael. 1 minute, I will quickly open my presentation. Right. Yes. And many thanks for all the organizers for the arrangements. I hope all of you behind the screens have enjoyed the deep diving into the project as much as I have this afternoon. So very good afternoon. My name is Anni Kaukoranta, and I come from the Ministry of Economic Affairs and Employment. I'm very pleased to bring greetings in between the great presentations on behalf of the ministry. Our ministry is really a growth ministry, I would say. Our goal is to generate socially, economically, and ecologically sustainable growth. Together with other ministries, we have been following and supporting and also funding the establishment of the National Center for Neurosciences from the very beginning. And we are very much looking forward to the day when the center is up and running. Today in this webinar, we have presentations of the 6 pilot projects that started quite exactly 2 years ago and that have received funding from the Ministry of Social Affairs and Health. I have to say that coming from the ministry and being a civil servant with a background in social science, it is very fascinating to learn about the very specific research projects and outcomes we have had this afternoon. Finland's competitiveness and well-being are built on competence, research and innovations. Finland's rise out from the emergency caused by the current pandemic and success in global competition in more general terms require the production of new knowledge, a high level of competence, as well as innovations that bring social benefits and added value. Finland has invested in health-related science, research and education, as well as the research infrastructures and public healthcare system already for decades. The recent research and innovation activities, like the ones we have heard today, are an indication that by working together, we are on a right track. But in spite of these investments, we still have room to improve, and we need more cooperation between higher education institutions, researchers, companies and private sector, and other R&D actors. And enhancing this type of public-private cooperation and partnerships is a key objective of our ministry and also the health sector growth strategy. The strategy was published in 2014 and continues to be implemented jointly by the ministries, research organizations, the business community, university hospitals, and other stakeholders. in research and innovation field. The current government is already actually the third coalition government that takes forward the goals that were agreed over 6 years ago. And we will very soon publish a new health sector roadmap that will set the agenda and actions for the next 4 years. Finland has a very good starting point to succeed as a leading country in competence and innovations. Companies are key research partners and engines of making choices for higher education institutions and research institutions as well. Research and networks must, however, be grouped into larger competence centers and ecosystems in order to strengthen, broaden, and increase the competence. Our ministry, the Ministry of Economic Affairs and Employment, the Ministry of Education and Culture, and the university cities together support the building of internationally active and attractive innovation clusters and the linking of thematic innovation networks to global value chains through the ecosystem agreements that are being negotiated currently. Well, in the health sector, nationally networked and strong clusters representing specific research areas such as neurosciences bring well-being and growth and attract international researchers, experts and professionals, as well as knowledge-intensive companies and international funding on the levels that no player would be able alone. The combination of our biobanks, healthcare registries, and the R&D-friendly regulation creates a very competitive environment internationally. Being able to rely on ethically sound legislation and research practices is not only important for our citizens, but a trustworthy environment is also a key enabler for the whole research community and the companies as well that do research in Finland. Digital health, personalized medicine, and scientific research in areas such as neurosciences are among the areas where we are today world class. Our objective is that we have established in the coming years the Cancer Center, Neurocenter Finland, National Genome Center, Drug Development Center, and at the same time we are also taking actions to enhance the collaboration between biobanks. The quality and level of the neuroscientific research is high, as we have heard today. A key opportunity lies now in further strengthening the cooperation between different universities, university hospitals, and other research institutions. Today, we can have a snapshot on this when we hear about the outcomes of the pilot projects. Moreover, opportunities exist in increasing collaboration with the industry and healthcare system as a whole. And major growth potential exists in this field. Success in global competition is calling for joining forces and enhancing collaboration. For example, that national actors join forces to create these types of centers of excellence and infrastructures with international significance and potential. And this is achievable when all the actors recognize their common interest and they work together. So this was my small greeting in between the presentations. Thank you very much, and I hope you enjoy the rest of the event. Thank you.
Okay, so thank you very much for the kind words, and we are really looking forward to collaboration with the ministry also in the future. So then I would like to welcome our next speaker, Professor Kari Majama from Oulu University and Oulu University Hospital, who is the coordinator of pilot concerning rare neurological diseases in Finland, especially Huntington's disease and genetic motor neuron diseases. So please, Kari, the floor is yours.
Thank you, Mikael, and greetings from the north. I will present results from Pilot 4 that deals with neurological rare diseases in Finland, and that was set up to be a collaboration between Oulu University Hospital and Helsinki University Hospital. Interest in rare diseases started to emerge at the turn of millennium, when France was the first country to publish an action plan on rare diseases. This took place in 2004, and a few years, 5 years later, European Union reported or published a recommendation regarding actions in the field of rare diseases. There are many definitions on rare diseases, and the European Union defines a disease rare if it affects fewer than 5 subjects in 10,000 people. The number of rare diseases is high. It has been estimated that there are more than 8,000 different diseases that are rare, and combined altogether they affect some 6 to 8% of the population. Most of the rare diseases are genetic, and the estimate is around 80%. In Finland, the EU recommendation led to the establishment of a steering group that prepared a national plan for rare diseases that was published in 2014, and it contained 13 proposals for measures aiming at developing research, care, rehabilitation, and social care. And in this slide, these 13 measures are shown in Finnish, and I have highlighted in red font those aims and proposals that, in my mind, fit very well to the scope of Neurocenter Finland, including registry, research program, strengthening international collaboration, expert centers, and systematic collection of knowledge. The Pilot 4 deals with 2 aspects of, or two examples of rare diseases. They are Huntington's disease and motor neuron diseases, and they combine the expertise gathered during the years at Oulu University Hospital and Helsinki University Hospital. And one of the aims was to evaluate possibilities to create a national register for rare diseases. First, a few words on Huntington's disease. It is an example of classical neurological diseases. The onset of the disease is in adulthood. There appears to be a pre-diagnostic phase when the symptoms are minor. They are cognitive and psychiatric most often, and then the diagnosis is made most often on the basis of the motor phenotype, although in some cases also the cognitive and psychiatric features can be marked. Huntington's disease was one of the first neurological diseases where the gene etiology was discovered. It is caused by an expansion of a 3-nucleotide repeat sequence in chromosome 4. And research has shown that when this repeat sequence is smaller than 26 repeats, the allele is normal, and when the number is 40 or larger, then it is definitely disease-causing. And in between, there is a gray area. Finland is peculiar in Huntington's disease in the sense that the prevalence of the disease is lower here than in other European populations. This is the table shows a recent systematic analysis on 14 studies on European populations, and it shows that the median prevalence is 5.7 per 100,000 population. But in Finland, the frequency or the prevalence is 2.1 per 100,000, so almost 1/3 of the European median. The interesting thing in the Finnish population is that the Huntington gene haplotypes differ in the Finnish population from that in other European populations, so that the haplotype A that is associated with the propensity of the 3-nucleotide repeat to expand, the frequency is lower in the Finnish population. And we found that the frequency of this haplotype, it is higher than haplotype C, and the expansion is more probable to occur on haplotype A background than on haplotype C background. The aims as far as Huntington's disease goes of Pilot 4 is to try to create a national register on these patients, both patients and carriers of the Huntington gene mutation, and to obtain experience on registers, what information is possible, what information is feasible to collect and with reasonable effort, and what kind of resources are required. And then for subsequent research, biobanking DNA and making way for molecular research. And so far, the data collection pilot has shown that there could be many data elements that could be registered. These were evaluated with the result that only few were accepted. And it became clear that comprehensive data can be obtained only if the inclusion is automatic and based on ICD code. And this, of course, would require that the system is integrated to patient information systems. Furthermore, reliable data can be obtained if the hits are then reviewed and verified before acceptance. Our previous experience on Huntington's disease has shown that we identified nationwide 400 patients on the basis of G10, the ICD code, but chart review revealed that only 207 of these, so 50% of the hits were correct. The reliability of clinical data depends on clinicians doing their daily work and depends on the accuracy of entries that they make. And this is the conclusion that we have reached. We came to the conclusion that a single generic rare neurological disease database is feasible. So it is impossible to create databases for each rare disease separately. There should be a single database that is generic to the largest extent possible. And the left part of the slide shows the common data elements that we sort of came to the conclusion, and it includes an example of patient-reported outcome, that is the 15D health-related quality of life measure, and in order to make the work amount as small as possible, it is necessary that we rely on these patient-reported outcome measures. And one further issue that came across during the process is that such a registry should be compatible with the European registry project. And in fact, as we speak today, there has been a European meeting discussing this European registry project, and it is coming to the same conclusions as we have done. Only core information should be collected, and a single database should be attempted. And the plan is to rely on the REDCap database platform and to create a system where the quality of care could be followed. And then this shows the elements that are being planned in the European registry. They are pretty much the same as ours, with the exception that they are planning to use the ICF classification of function, which is rather complicated in my mind. The other aspect of Pilot 4 is motor neuron diseases that includes spinal muscular atrophy, a pediatric disease, amyotrophic lateral sclerosis, an adult disease, and various hereditary motor neuropathies, including Losmon, a disease discovered in the Finnish population and that appears to be rather frequent among the Finns. Whether it is part of Finnish disease heritage, it's not yet known. And the aims in this MND part was to develop an infrastructure, a registry for diagnosis and follow-up, identify biomarkers, and study disease mechanisms. And this is a chart showing the content of the MND registry. There are 800 people already, but detailed information is on 277 individuals. Gene diagnosis has been achieved in some 45% of these 277 subjects. The Losman mutation and GDAP1 and PMP22 causing motor neuropathies, and SMN1 causing the SMA, childhood SMA, are the most frequent hits here. And this registration has already enabled the identification of one new disease-causing gene as well. A biomarker study has been focused on Losman. It's caused by mutations in CHD10 gene, and the PG66V is a frequent mutation in the Finnish population. And a metabolomic study on these subjects have enabled the identification of new markers, new biomarkers that enable follow-up of the disease activity and may enable to follow treatment effects. And the third line in the study is use of induced pluripotential stem cells that have been established using skin fibroblasts from Losman patients, and these studies on cultured motor neurons has enabled the finding that the mutation affects gene expression and mitochondrial function in motor neurons. What are the benefits brought about by Neurocenter Finland to this project? The evident one is the fact that it led to a closer national collaboration between centers that work on rare neurological diseases, and this collaboration further led to the application to become a member in the European reference network for rare neurological diseases. The application round was 1 year ago, but the COVID epidemic has delayed the process, so that at the moment we don't know the fate of our application. The application was a consortium including both adult and pediatric neurologists, and it consisted 3 university hospitals, Oulu, Helsinki, and Tampere. The pilot project has brought important experience on registers, especially on the methods in data selection and collection, evaluation of the features of high quality register, and also exploring the technical solutions from both academic and commercial perspectives. and also it has enabled research on mechanisms of motor neuron diseases. And other experiences from this pilot include a collaboration with patient organizations. It turned out to be a very valuable route to identify patients with rare neurological diseases, and definitely this route has not been fully employed in the past. In addition, this pilot project enabled closer collaboration with pharma industry, and this has led to various kinds of attempts to further the activity in the field. And the third experience was that altogether societal activity in the field of rare diseases has increased. And these are the participants of Pilot 4. I would like to thank their participation and acknowledge their contribution during the pilot, and also I'm obliged to Neurocenter Finland for making this pilot possible. Thank you.
Do we have now any questions from the audience? If not, then I would ask actually one question. How do you see the role of genomic data in the rare diseases or rare neurological diseases registered in the future? So do you think that it would be important to get that data from FinGene, for example, or from biobanks, and in that way augment the content of the registry?
Mm-hmm. This is a very good point, and we have discussed it quite a lot. Biobanking of samples from patients with rare diseases and biobanking exome sequences, full sequences of these patients, and making them available is extremely important for the development of the field.
Okay, so thank you. Then I think we move to the next speech. Coordinator Jonas Haapasalo from Tampere University Hospital will present their pilot on clinical molecular genetic and translational research on brain tumors. So, Jonas, the floor is yours.
Thank you. Can you see my presentation already?
So Jonas, we don't see the presentation yet.
Yes, can I have the right to show the presentation? It seems that there's some sort of technical problem now.
Okay, so
yes.
Now it should be working.
Okay, now it's better.
Okay, thank you.
So perhaps you can see my presentation soon. Okay, can you see my slides there? Okay, my name is Jonas Haapasalo. I'm a neurosurgeon from Tampere University Hospital. And I apologize
Jonas,
that...
we don't see the slides yet, so...
All right. Just a sec. How about now?
Maybe it's a wrong screen. It looked like a directory.
All right. Sorry about the technical problems here. How about now?
Oh, it's still the same screen.
Okay, let's try again. Can you see it right now?
Yes.
Okay. Sorry about the delay. Sometimes it's hard even for a neurosurgeon to get the lines working. But like I said, my name is Joonas Haapasalo. I'm a neurosurgeon from Tampere University Hospital. I don't try to turn the video on because if you now can see my presentation, it's clear enough. And actually, because I had on-duty call yesterday night, it's far better if you see my smiley face on the right-hand side. That's actually a nice bonus because it's something like 5-year-old picture for me. So I'm talking about the pilot number 5, which is about the brain tumors. And first, some background about the disease. So you all might know that there's approximately 1,000 new cases, new patients per year in Finland. So it's not considered as rare disease. At least neurosurgeons, neuro-oncologists, and neurologists deal with this disease almost every day in the clinical practice. It's a very heterogeneous group of disease and different kind of tumors. On the right-hand side, you can see 2 pictures from the World Health Organization classification of tumors. And there's a rapid change and development on the diagnostics. So when they operated the classification from 2007 to 2016, lots of new nice things occurred. So actually, there's lots of things dealing with the genomic changes and even DNA methylome-based data regarding the diagnostics. Sadly enough, still most of the brain tumors, especially the malignant ones, have very poor prognosis. So one of the key problems of ours is that the current treatments, which are neurosurgery, chemo and radiotherapy, aren't enough. And of course, there's some promise and hope for developing better treatments, even targeted therapies for malignant brain tumors. I have to underline that actually in the pediatric population, it's the second most common tumor type and the leading cause of mortality and morbidity in Finland. So we are dealing with a big problem in the pediatric population as well. So the aims of the pilot. Within the same slide, you can see the aims and perhaps some results, or at least some numbers, because I would say that neurosurgeons are concrete people. I would like to deal with things that I can touch with my hands. Well, the numbers I cannot touch with my hands, but at least I can show them now at the beginning. So the first aim was to build a collaborative network between all 5 Finnish university hospitals and universities. And I'm very glad to announce that it was rather a simple task. So there was lots of motivated clinicians who wanted to join the pilot. And we established the network of all 5 university hospitals in a way that actually the site coordinators, all of them are neurosurgeons, basically most of them my friends. And the thing that we wanted to establish is this sort of informed consent with the ethical permit, of course. And we recruited the patients with this consent from each university hospital. So from Tampere University Hospital, which was kind of the example how to recruit patients, we recruited 250 patients during the pilot time. And nicely also the other centers recruited more than 100 patients. And we gathered some clinical data and follow-up data first. And of course, within the neurosurgical operation, we collected tumor tissue, either with biobanks or with local tissue collections. And we also collected blood samples for the genetical analysis, for example. And from some patients, we also obtained cerebrospinal fluid, approximately from 80 patients. And we also wanted to make some registries, and one of the key things in this project was to evaluate if we could use some data mining tools and use them in patient records. We have some preliminary results from those studies. And of course, because this pilot was partly translational, we wanted to establish collaboration with different national universities, but also with international brain tumor centers, such as University of Heidelberg, Hospital for Sick Children in Canada, and University of Sydney. But I will tell something about these projects as well. So this picture is just to remind that obtaining the brain tumor sample isn't always straightforward or simple. So basically, always we have to do either biopsy or craniotomy and then resect the tumor. So it's a bit risky business, and part of this project was to evaluate if we could obtain some samples in addition to normal diagnostic samples, which of course play the crucial role and are the most important steps with the diagnostics. And I have to say that it was nice to see that neurosurgeons performed operations and took some samples either to biobanks or tissue collections. And kind of the baseline of the study was that if there was some extra need for diagnostic samples, we would give, of course, the research samples back to the clinicians for the diagnosis. And actually, the first thing that we did in this project was establish a strong clinical collaboration with Turku University Hospital. There we did some nice collaboration with Jussi Posti-Villevuorinen and also Jussi Sipilä, who actually works in University of Eastern Finland now. And we gathered clinical databases in kind of a hard way. So the scientists looked for the patient records and did this job manually. So we built two similar databases of approximately 1,000 brain tumor patients, and we had some clinical follow-up data for 15 years. So this project was retrospective, and we gathered some epidemiological data about primary symptoms and leading symptoms in gliomas, which are the most common primary brain tumors. And we could describe the leading and primary symptoms of malignant gliomas in South Finnish population. And even with the modern diagnostic criteria, which include the molecular diagnostics, we can use these symptoms, for example, leading symptoms that haven't been studied yet. They could be used as prognosticators even in molecular era. That means that in multivariate analysis, these characteristics came up. So you could use the clinical symptom as personalized tool to evaluate patients' prognosis. on the right-hand side, kind of evidence that if a patient has motor symptom, the prognosis is rather bad. Then some words about the Tieto Data Explorer or the Data Lake project, which was part of the second part. So because it's quite hard and time-consuming to go through all the patient files manually, we wanted to study the automatic data mining of patient records, and we used the patient data from Tampere University Hospital, which was then transferred to the data lake of Istek Oy. And then we had a nice collaboration with Tieto Oy, who developed a patient data explorer. And this has been used in other projects than brain tumor projects, so we didn't have to start from the scratch. And then we used the artificial intelligence and trained the program, and we obtained some data from the algorithms. The current situation is that the algorithm has been trained with some clinical features such as epilepsy status and tumor molecular status, and this data now has been compared to the data that we have from the existing databases. We have to do some training of the algorithm still, but the first results show some promise, and perhaps we could use this sort of algorithm and program for data mining for biobanking purposes as well. So it's interesting. Then something about the molecular genetic part of the pilot. We started collaboration with the University of Heidelberg in Germany with Professor Christian Beitler. They have been studying epigenomics of brain tumors for a long time, and they are pioneers in Europe in this field. And in this pilot research, a pediatric hemato-oncologist, Nina Forss from Tampere University Hospital, has been the key player because her material consisted of both brain tumor samples and CSF samples. And from the pictures A and B, you can see some chromosome epigenomic analysis that they performed in Heidelberg. And from the panel A, you can see the tumor chromosome copy number analysis from freshly frozen tumor samples taken from the patients. And from B, equally chromosome copy number analysis from cerebrospinal fluid. And it seems that we could do this sort of liquid biopsy. It's easily done by lumbar puncture. And when we obtain the CSF, that's liquor, we could make the analysis with the epigenomic methods, and actually before touching the tumor, making the craniotomy, make the diagnosis. Here is an example of plexus carcinoma. So this needs further investigations, and we are gathering more material actually from all university hospitals now, hopefully. And we have already started collaboration with Tampere University Hospital with Professor Nykter and Kirsi Kramberg, who are evaluating the possibility for next generation sequencing in cerebrospinal fluid. Then some words about the collaboration with industry. So translational research contains the projects of EINOS. That's something that we have been doing with a company called Olfactomics, and Professor Niku Oksala and Professor Antti Vehkaja-Oja from Tampere University. Basically, this machine does the same thing that the dogs do at the airports when they sniff the corona. So we want to have this sort of odor fingerprint that we could use in analysis or diagnostics of brain tumors. And last year we published one proof of principle article about the DMS, that's the differential ion mobility spectrometry sensor. The prototype is shown in the right-hand side picture. So this has been tested in laboratory setting. Basically, you can burn the brain tumor tissue either with the surgical knife or surgical bipolar pincers that we use in routine practice. Then we collect the smoke, and then we analyze the smoke with this machine. And because it's already useful and shown to be useful in diagnostic purposes when you consider histological diagnosis, we are aiming to study further. So if this machine, eNose, could detect, for example, genetic mutations, chromosomical aberrations, or some other features of brain tumors. And we have studied project both in both adult and pediatric populations. And the Neurocenter trial has been key instrument to collect these rare tumors. Here you can see just some examples how it works. So we use linear discriminant analysis and artificial intelligence to classify different tumors. And in this proof of principle article, we actually showed that low-grade gliomas can be compared or distinguished from the high-grade gliomas. Those are glioblastomas, and the accuracy would be something approximately 94. So I assume this would be kind of nice clinical tool. And at the bottom left-hand side, you can show the picture from the operation. So actually, I'm using those pincers to coagulate the blood and to prevent blood loss. And if we could just adjust smoke suction in the operation theater, then we could analyze non-invasively the whole thing with this machine. And actually, Nikuoxala and olfactomics have been using this already in breast cancer. So in clinical setting, we are aiming to go to the OR someday. There has been other projects that needs to be started. We have been struggling with GDPR. So even though we have the informed consent and ethical permit, it has been a bit tricky, or at least the bureaucracy has been a bit slow. But this is just some example that we could do nice collaboration with Turku University Hospital and Turku University. There's a laboratory of Maija Holmén in Turku University, and they are studying Clever-1, which is macrophage expressed receptor, and that suppresses the macrophage activity and is highly expressed in GBM. We have already analyzed some tissue microarrays or tumor microarrays and screened Clever-1 in different brain tumors, and it seems that this might be a nice candidate for targeted therapy. And the plan is, when the bureaucracy is done, to get some tissue samples to the laboratory of Holmén and made some organoids, and they can perform the experiments then. Anti-Clever-1 medication, by the way, has been already tested in other cancers, and there's also industrial collaboration with Faron Oy and Juho Jalkanen, which is hopefully taking place in the near future. So to sum up my presentation, the benefits of the Neurocenter trial had been, of course, the establishment of nice collaboration network between different university hospitals.
Okay, so I think we have lost connection with Jonas for some reason. So Jonas, do you hear us?
Okay, can you hear me now?
Yes, now we can hear you.
Sorry about bad line, but the collaboration has been the key. So the formation of the network that I presented here, that I present in this slide, is key. And also we have found new translational ideas, and we have established collaboration with industry, and that's due to the fact that Neurocenter network has been working very nicely. So I'm very grateful for the opportunity to be part of this pilot trial and work as a coordinator, and hopefully we will continue with the collaboration that has been established. Thank you very much. Hopefully, if you still can hear me, I can perhaps answer to your questions, and then if not, you can at least send some email.
Okay, so thank you, Jonas, for the great presentation. So now there is room for one question. So do we have any from the audience? Okay, so if not, I would like to ask one question. So I think that you have done excellent work in establishing a national collection of fresh tumor samples, which is not an easy task. And I would like to hear about your experience so far. So what has been the most difficult part of this job, and what would be the advice for a researcher in other fields of tumor or cancer research for starting this kind of national collection?
I think the hardest part has been to establish good talk with the bureaucrats, because this has been kind of a difficult task for us all. So most of the neurosurgeons have used the tissue collections, basically freezers of the neuropathologist. It would be very highly beneficial to automatize this or use the biobanks. And I think we have been using biobanks in this project, and this far, I think after 1 year of lots of paperwork, we are in current situation. So hopefully the tissue that has been biobanked can be used in many occasions, and also the tissue collections. But I think these difficulties comes from the GDPR and the legislation that has been quite new in this field.
Okay, so thank you. And then I think we will move to our next speaker, Laura Airas from Turku University and Turku University Hospital, and she will present her pilot on multiple sclerosis from molecular mechanisms to personalized care. So Laura, the floor is yours.
Yes, thank you very much, and good afternoon to you all, and thank you for bearing with us until the very end. So I will be describing the pilot 6, which concerns multiple sclerosis. I'm based in Turku at the Turku University Hospital and the University of Turku. So the pilot 6 was also divided into 2 parts, and first of all I will talk about the part 1. So here the collaborators are, in addition to our group in Turku, there's Susanna Narkilahti, the PI from Tampere University. She's a stem cell researcher. And then I must highlight also the importance of Dr. Sanna Hagman for this project. She's shown in the picture. So Sanna did her PhD at Tampere University with Professor Irina Elovaara on multiple sclerosis. And we have been toying with the idea for a long time that we should really start a collaboration together. And I must say that it was this Neurocenter that then triggered really this collaboration between us. So the aim of the project is to shed light to the pathological mechanisms that contribute to progression in MS disease. And our approach is to explore the effect of MS-associated risk genes on the phenotype of the pathogenic glial cells. So you are probably all aware that that in the field of multiple sclerosis, the greatest unmet need is the lack of efficient treatment for progressive MS. And this leads to great individual suffering, of course, with the loss of ability to move and impairment in cognition in this patients, but also to vast economical costs for the society. So in relapsing remitting MS, which is the most common MS type, the active inflammatory lesions are really the basis of the pathology. And here the adaptive immune system has a significant role. But as the disease advances, over 60% of the relapsing remitting patients convert to secondary progression, and this is where the disease pathogenesis changes. So there's not so much trafficking anymore from the periphery into the central nervous system of the adaptive immune cells, but inflammation becomes trapped within the central nervous system. There's this compartmentalized, diffuse, smoldering inflammation, and this includes microglial activation. There's oxidative stress, mitochondrial dysfunction, and all of this then leads to atrophy and disability. And this is much more difficult to target therapeutically. It's fairly easy to block the immune activation in the periphery, but actually to target the innate immune system and the neurodegeneration in the central nervous system, that's difficult. So as in many other chronic complex diseases, in MS, also a GWAS or genome-wide association study was done. And it came out with a high number of susceptibility genes. So over 200 risk SNPs were identified. So these would increase the susceptibility to get MS. And so I stress here again that these are not mutations, right? But common allelic variants, risk variants that increase the susceptibility. And as many as 20% of these risk genes are associated with NF-kappa B. So this is the pro-inflammatory transcription factor, which is of course highly interesting. Let's see. And there has been some functional studies looking at the effect of these risk alleles on lymphocyte action and lymphocyte phenotype. When I was working at Yale University during my sabbatical about 4 years ago, already then I was exposed to this idea that these risk variants could also cause a disease risk state in central nervous system cells. And a good collaborator and friend, David Pitt, he was studying this at the moment and at that time, and his approach was to investigate how these risk variants would affect glial cell function in vitro. And here he was already using the induced pluripotent stem cells, and with a thought that some of these functions, altered functions, would be relevant to neurodegeneration and contributing to disease progression. And actually this work has been published in a nice paper where the cell of interest was astrocytes. So we have raised the question then whether these MS risk variants actually would lead to increased microglial and astrocyte activation also in vivo. And here PET imaging of these glial cell types gives us a tool to actually address this question. So what we have been now doing in collaboration with the Tampere group, so we have genotyped MS patients, and we have chosen patients with either the risk variant of these NF-kappa B related genes or a protective variant. And we have collected blood from these respective patients, and at Tampere, they have been then generating iPSC lines from these patients, and then these pluripotent stem cells will be then differentiated into microglia and astrocytes. And then it can be tested in vitro whether there is a difference in the cellular phenotype and function regarding whether the individual who the cells are derived from was carrying a risk or a protective allele. And then at the end, we can then do the PET imaging for microglial and astrocyte activation and see whether this holds true also in vivo. And then this can be, of course, and used as an imaging biomarker when evaluating MS patients. So our thinking is that the genotyping of patients and understanding the glial activation mechanisms is a step towards precision medicine, towards personalized medicine in MS, because this would enable us to identify those MS patients who particularly need treatment to target the disease progression. And this is also an aid in drug development for progressive MS. So again, with this Pilot 6 project, it was the Neurocenter initiative really that triggered us to create this collaboration. And during the course of the project, this network has grown also beyond these original collaborative centers and collaborators. So just a few words about funding. So initially the part 1 of the Pilot 6 was funded with 100,000 euros. But especially Sanna Hagman has been extremely successful in securing future funding for this project, which is really, of course, important because the cell culture experiments are still coming, but now we know that we have good funding for that work. We have also got new funding to secure continuation of these studies here in Turku, and just 2 days ago we learned that Academy of Finland is actually funding this In Flames flagship program in Turku, and we're also part of that. So that's very nice. But there are also still pending grant applications with collaborators associated with this project. So then a few words about the Pilot 6 Part 2 project. So here, this part of the project was led by Professor Pentti Tienari at Helsinki University Hospital, and all university hospitals in Finland are taking part. And the initial purpose of the project was to design a study for an essential drug comparison. So the story is that there are 2 lymphocyte depleting drugs available for use in Finland at the moment. The older and cheaper one of those drugs is rituximab. So the retail price of an annual dose of this drug, it depends a little bit on the manufacturer, but it's between 2,500 and 6,000 euros. Now there are also these biosimilars of rituximab available, so this pulls the price down. This drug only has an indication for treatment of, for example, rheumatoid arthritis, so it's not indicated for MS. So all use of rituximab is for MS is basically off-label use. Now we also have this newer and more expensive B-cell depleting drug, ocrelizumab, and for this drug, the price per year is nearly 24,000 euros. And this B-cell drug has an indication for treatment of MS. And by now, so neurologists all over the world have when the good results on the efficacy of the B-cell drugs in MS came out, so the neurologists all over the world started using rituximab for treating MS. So there's plenty of real-world data available on that, but there's actually no formal studies comparing rituximab and ocrelizumab. And considering the price difference and the identical mode of action, we figured that it would be very important to compare these two drugs head to head, both their safety and their efficacy in the treatment of MS. So this part of the study clearly also has some health economical aspects to it, and very, very clinically relevant questions. So we actually published our experience of the use of rituximab in treatment of MS at our university hospital. And it is an incredibly effective drug. So in the figure on the right, you can see that before initiation of treatment, patients were experiencing relapses, but then during the first and second year of treatment, basically no more relapses at all. Okay, but it became evident very early on that this head-to-head comparison study couldn't be conducted as planned because of some budget issues related with the hospital-administered infusion drugs, and the study plan was altered, and the Pilot 6 Part 2 had to go with the Plan B, and the resources were directed towards creating a national system for collecting and storing blood samples from MS patients for research use. And there have been some successes with this. So Professor Pentti Tienari, who was the coordinator of the Part 2 of the project, he organized the ethical permissions covering all participating centers. The sample collection is planned to continue beyond the pilot project's finish date. Each of the centers is looking after organizing the local sample collections as well as the processes involved in the research permits. The samples are stored at the 5 Finnish biobanks, and they are available for all MS researchers or any researcher according to the current biobank legislation. So also the Part 2 of the Pilot 6 has secured quite good future funding. There has been some challenges along the way, as has been alluded before in several of the talks today. So the amount of paperwork has been quite heavy. There's a surprising amount of still heterogeneity in the need and nature of the contracts and processes between different centers. And this has clearly slowed down the start of the project, and it's a clear obstacle in creating smoothly running national collaboration. But of course, now we're again wiser in how to do this in the future. And I also hope that maybe Neurocenter Finland could do something to sort of smooth out this path a little bit. At the end, I must say that support from Neurocenter Finland will be essential also in the future. And as always, the functional and stable funding is the foundation for successful and high quality research. I thank you for your attention, and I think we're happy to take any questions. I think Pentti Tienari is also now here on the lines.
Okay, so thank you, Laura, for the great presentation and excellent work in translating the knowledge from bench to bedside, so to say. So now we have time for questions. So do we have any from the audience? Okay, so I have actually one question. So Laura, you mentioned that collaboration would be important also in the future in the form of new funding. But I also have another idea, or actually a question. So how do you see the possible collaborations with the patients or patient organizations now or in the future? And is there something that we could do together in that area?
Definitely. So I think when we are collecting the biobank samples, I think definitely the patient organizations could be useful here in getting the message through to patients that everybody's sample is important and needed, and this will sort of would increase the numbers of the samples stored in the biobanks, which would then be available for everybody to use. So a very, very important, yes, the third sector.
Okay, so thank you. So does anyone has any comments on Laura's speech or other speeches of today? So if not, then I will just very briefly wrap up. So we have had excellent day today, listening to wonderful achievements in Finnish neurosciences. So it has been really impressive to see so much world-class research being done here in Finland. And I think that there are also great examples of working together. So there's a lot of networking behind and also a lot of work behind all these presentations and results that have been achieved. And Neurocenter Finland is really happy and proud that we have been able to be part of these pilots. We have been able to advance them maybe a bit further. But the story continues, so we would like to welcome all of you as part of Neurocenter Finland because together we are much more than a sum of individuals. So I wish you a nice rest of the day and stay healthy.